Be right back. Nope, I'll put the screen up and then I'll be right back. I'm really just trying to minimize this. There we go, for real. 483115. Okay, now for real, I'm gonna go backwards. Back. Get it on your laptop. Could you put it up on your laptop?
No, it doesn't.
Oh,
Yeah.
you're like, I'm doing it.
Give a couple minutes, just make sure everybody gets signed in. All right, if you're online, please just double check that you're signed in. We're gonna get started here in just a second. All right, we're gonna go ahead and get started with our Kahoot. It shouldn't take the whole time, the whole 2 hours. It should only take about an hour and a half, maybe. So hopefully it won't be too, crazy for you guys. I'm not gonna call on anybody individually or anything like that, so no worries, but just be prepared for the discussion. Why am I not finding there? So I'm just marking everybody as present who's online. Oh, there's Kate. Okay. All righty, so let's go ahead and get going. I keep seeing people's names pop up. One thing on my thing. She does for my class. I nearly. That's it. Making sure I'm not missing anybody. I'll double check it later too. Okay, so let's go ahead and get started. We start off basic, really basic actually. All right, so one of the things that we need to be aware of, I'm going to sit down like through parts of this too. I'll stand up if I need to draw on the thing, but otherwise I'll be sitting down because I had to get up at 4 this morning for Pilates. But I mean, not that I don't love my Pilates, but it just was an early day. It was a lot of hip workout. So you're gonna bear with me for this one. All right, so anyways, safety, the basics for lab safety. So a little bit of a preface here. You must have your lab coat and your closed-toed shoes for lab, for the lab exam. You will be counted off points if you don't have them. So just be sure you have your lab coat and your closed-toed shoes that day. If you're missing one, then you get 2.5 points off. So up to 5 points off if you don't have closed-toed shoes and you show up without a lab coat. I will loan you one so you can take the lab exam, but it'll cost you points in order to borrow one. So be aware of that. You do need your own lab coat for that day. So that's part of our lab safety knowledge, but you also might get asked questions about general lab safety knowledge. And it's not going to be anything complex or difficult. It'll be questions like the next one that we have. Where should broken glass be disposed? Now, I wish I could promise you that the whole test will be this easy, but it won't, of course. So, but yes, easy enough. Broken glass gets disposed of in the sharps container. In fact, even intact slides get disposed of in the sharps container. If it's in the biotrash, we're assuming it won't poke holes through the biotrash whenever we are collecting it and everything like that. So that's the issue there. If it's been contacted, contaminated living bacteria, then it will go in the biotrash unless it's made of glass, then it goes in the sharps container. So easy enough. Question, stains. Whenever we're getting into our stains, so I always forget to start out with this. First of all, I want to show you guys something. In your Canvas course, if you go to today's module, which would be week 8, I mean, don't hold your breath because Canvas is very slow lately. But once you finally get that pulled up, anytime. Okay, week 8. And they have the dates there, again, in case you aren't much of a modules person. I would hope we would know by now, but just in case. Okay, you see where I clicked on this, right? At the very top where it said lab midterm study sheets or whatever. This is a compilation of basically a summary of each of those labs. I'm not saying it has every single thing you could ever possibly want to know about them, but it summarizes the main ideas of each of those labs on one page for each one. Like I said, the main ideas. And so that's there. And I encourage you guys to make your own version of these. But the real reason I'm showing you this, number one, this has only midterm stuff on it. All right, number one. Number two, it has this thing at the very bottom. This is my appendix. It's the summary of the stains. The stains. So the motility wet mount isn't on here because it's not a stain. But if it had a stain involved in it, then it is on here. So simple stains, gram stains, acid-fast stains, capsule stains, and endospore stains are all on here. The purpose of each one, whether we heat fix before, then the primary, mordant, decolorizer, or counterstain for each of those. So those are the steps. I don't care that we put it on there for 1 minute. I'm not going to test you on that ever. But I do care that you know what order we put them in and the purpose of each of those steps. So this doesn't really cover the purpose of every step. This is the gist. This is your big picture idea for these stains, right? So for example, for the gram stain, we're wanting to ask, does it have a thick or a thin cell wall? We do heat fix before we do it. Our primary stain is crystal violet. If you have a thick wall, the crystal violet gets trapped in there. So that's not on our little mini cheat sheet here, but we should know that. Then iodine is our mordant. So I highlighted mordant here because that's a whole other term that we need to be aware of. A mordant is anything that fixes your primary stain into place. And so in the case of a gram stain, our mordant is iodine. Iodine fixes crystal violet that got trapped in the thick wall. Then we have ethanol is our decolorizer. Decolorizers always rinse out whatever didn't get fixed in place by our mordant. And then our counterstain is a safranin. Generally speaking, our counterstain is always going to be there to color decolorized cells. So we can almost think of our counterstain as generally just another simple stain. Simple stains are just there to provide contrast so you can see the cells. And most of the time our counterstain, that's the purpose of them. So we can just see it, right? And so that's all here. What does the positive look like for each one? What does the negative look like for each one? Anything special information-wise? Generally, the gram stain is where we start for identifying organisms, bacteria. If you know it's a bacteria, usually the first step is it gram positive or gram negative? And then we'll do the other stains after that. But yes, so any special information stuff is there. Like I said, it's not completely comprehensive, but it's a pretty good starting point for the basics for the differences between our stains. Simple stains are there basically for us to allow us to see the cells and then any, defining characteristics of the cells like cell shape or arrangement. But gram stains, acid-fast stains, capsule stains, and endospore stains, they're differential stains. So they tell us the difference between things. So you're either positive or you're negative for those. You're not positive or negative for a simple stain, right? You're just seeing your stuff there. So anyways, that is there as a reference point for everybody. That can be helpful. And you can use that while we're doing our Kahoots if you want to. I have no problem with that. But again, on the test, you're not going to be given any cheat sheet information or anything like that, right? Okay, so that's a good reference point for our steps and the order of the steps, because generally speaking, again, generally speaking, for stains, you're usually going to start with your primary and follow with a mordant if your stain procedure has one, and then a decolorizer, again, if your stain procedure has one, and then a counter stain. Purpose of the primary stain is really, this is the characterizing one. This is where we're going to determine our positive and negative. The mordant is to fix the primary in place. The decolorizer is to remove unfixed primary stain. And then the counter stain is to basically give contrast to decolorized cells. Make sense? That's the gist of all of our stains. Okay. So we should know this purpose of each stain for every single staining procedure. So if we're talking about our acid fast stain, so really quick, coming back to our reference. If we're talking about the acid fast stain, primary stain is carbofuscin. Why are we using carbofuscin? Because it is lipid soluble. And we'll talk a little bit more about that information as we get into that one. Why we need why do we need a lipid soluble stain in this instance? Because we're trying to stain glycolic acid wax, and wax is a lipid. So for example, so know what's going on in your stains. It's not enough to memorize the steps. Don't care about how many seconds those things are on either. I'm not going to test you on that. I do need you to know the order. I need you to know what is your primary, why we're using that as our primary, right? And generally speaking, our counter stains are chosen because they contrast visually with our primary stains, right? So anyways, let's get into those. Be aware of what positive and negative will look like and what would happen if you skipped a step. I'm not going to ask weird, obscure skip a step questions, but if you skip the ethanol step in gram stain, for example, that's a big change in what your outcome is going to be. So those types of steps, I might ask some more information about. All right, so let's get into our questions for the stains. Select all that apply. What is the purpose of heat fixing? Okay, so the purpose of heat fixing is to adhere it to the slide and to kill it. The reason stain it isn't the right answer. The heat fixing itself doesn't stain it. It does prepare it for the stain. So you're not wrong if you did put that, but I'm not asking if it prepares it for staining. I'm asking if it stains it. So just also be sure if you're ever taking one of my tests and you're like, do you mean, does it actually stain it or does it prepare it for staining? You can always ask clarifying questions about stuff like that on the test. So just FYI, heat fixing itself doesn't stain it, but it does adhere to a slide and kill it. And we often use it to prep our slides before staining. Not always, right? So you remember which staining procedure we don't heat fix with? Okay, capsule stain, beautiful, perfect. So we'll be getting to that, of course. All right, next question. I'm gonna leave this up here for a little bit while we're doing our stains. What is the counter stain used in the gram stain? The counter stain for the gram stain is safranin. Safranin. So gram stain goes crystal violet, iodine, ethanol to decolorize, and then counter stain with safranin. All right, the next question is going to ask about the color. So what color will a gram-negative bacteria be after the gram stain? If you do it correctly. All right, so gram negatives will be red. Safranin is a red stain. Gram positives will be purple. Positive purple, PP. Negatives, to me, red is a negative color, and that's how I remember the gram stain specifically. Since that's our big bread and butter one, we definitely want to be the most familiar with the gram stain. Here's something I was going to say about it. Okay, one really quick couple of kind of note about the gram stain. The idea is that in your gram positive versus your gram negative. In your gram positive, you got this thick wall, and it's got this scaffolding in it. And the negatives have a thin wall, real thin. And they have an outer membrane and an inner membrane. They both have inner membranes, okay? And if you add crystal violet gets stuck in this scaffolding. Now, it'll stain, it'll associate like a regular stain procedure with the membranes. But then whenever we add ethanol, that'll get rinsed out. Whereas the iodine, which was our mordant, we use orange for that, or brown, kind of helps fix that purple in place. There's nothing really to fix in our gram negatives that was stuck to our membrane. There's nothing really to fix it in place. So then whenever we decolorize with ethanol, we're left with colorless cells on our gram negatives. And so the idea is the safranin will come back in as a stain and be associated with those membranes. This brings us back to that concept that we learned about in the very beginning with our staining, which was the idea of simple stains. So stains in general. You can either stain the background or you can stain the cells. Stains that stain the background versus cells. Does anybody remember what those stains that stain the background are called? Negative. Negative stains. These are also known as acidic stains. Stains that stain the cells are known as positive stains. They're also known as basic or alkaline. When you think of an acid, you think of it donates H pluses, plus something else that has a negative that it was attracted to, right? This negative that it was attracted to is the actual stain molecule. So negative stains are acidic, and then positive stains are alkaline. Why is this important? Because the surfaces of cells are made up of membranes. And membranes are just a lipid bilayer. Look familiar? I hope so. All right, and what are these little heads that I drew here? Phosphates. Look at all those oxygens in there. Phosphates are negatively charged. Negative attracts positive. So positive stains are attracted to the negative surface of the cell. That's why they stain the cells. Just a little chemistry rebrief on why we call them negative and positive stains. All right. Okay. So that's what will happen with safranin. Safranin is a positive stain. It stains the cells. Most of our stains are positive stains. Most of them are. We have one that's really important for another specific procedure that is a negative stain that we'll talk about in a little bit. All right. Any questions about this stuff yet? Okay, that's fine. We'll leave it up for now. What is the first stain used in the capsule stain? Different staining procedure now. All righty. The first stain used in the capsule stain is Congo red. Congo red. Next question is going to get into the nature of that stain. Because the whole point of the capsule stain was to visualize the capsule, right? The capsule itself didn't stain, didn't. So there's a way that we had to visualize it. I'm giving already too much away, I feel like. All right, choose all that apply. What type of stain is Congo red? I've already given a little precursor into what it could be up above. Congo red. Alrighty, beautiful. Most of us got this one. That's okay if you didn't. Of course, you're going to get it on the test, I'm sure. So Congo red is a background stain. We're going to stain the background and the cells, but Congo red stains the background, and therefore it is a negative stain also. Huh? Beautiful. Does anybody remember what our secondary stain? So in the capsule stain, we didn't properly use really a counter stain. We used a second stain, and it actually changed Congo red from red to purple. So we used the Manivels stain. Yes, the Manivels secondary stain, and that turned our background from red to purple and made it so we could see our cells too. As a secondary stain, so it was a little bit different. All right. All right, in the image is going to be a pure culture. What stain is pictured? All right, so first we kind of need to understand that a pure culture is just one species of bacteria, just one species, and it's a pure culture. The second thing that we need to understand is, obviously the staining procedure, which most of us got. This was the endospore stain. But how did we know that? Well, first of all, we know it because it was green and red, and none of our other ones were green and red. So we knew that. But also we knew it because whenever we looked at the image, we see different kinds of structures here. If this is a pure culture, we will not have a mixture of bacilli and cocci. That's because these are not cocci. These are endospores, which are a different structure. They're not cells. Those are not cells, not the round guys. The rods are cells, technically. Okay, so this is where we have to get into a little bit more understanding about this stuff. An endospore is dormant or inert and resistant. It's just lying in wait. Resistant. Whereas the other word, and I'm going to ask you guys in just a moment, is the active and multiplying version of the cell. Anybody remember what the term is for that? Perfect. Vegetative. All right. All cells have a vegetative form. All of them do. All bacteria, all cells in general. All cells will have a vegetative form. In bacteria, the ones that can form endospores will be Bacillus and Clostridium. It'll be in their name. This is the genera, which is the plural for genus, their genus name. So remember, species names consist of a genus and a specific epithet, and that two-name thing is the species name. So the genus portion of their name will have Bacillus or Clostridium in it. Okay. All right. We also happen to know that these guys are gram positive. It's something we know about them that we also need to know for both the midterm and the Unit 2 exam. So don't forget about that. I guess we don't really cover that in Unit 2. That was a Unit 1 thing, right? That was a Unit 1 thing. Okay, anyways, definitely lab midterm then. Any other thing I want to say about this? We're going to get into more questions about it soon, so I don't want to give too much away. All right, let's move on. So then what do we call the red structures that were in the image? It's the same image, the red structures. All right. So the red structures are the vegetative cells. The green bubble shaped things are green on the inside, or the little green parts of the cells. Those were our endospores. So we see here how we have some of these like bubble shaped guys that are filled in with green. Those are proper endospores. But you see how some of these rods have green at the end of them? Those are the vegetative cells that are forming endospores, and they have their own term for them. Vegetative cells that are forming endospores, we call them sporangiums. Okay. We learned about that in Unit 1, but hey, we're coming back to it here too. Okay, any questions about that? Anybody remember what the primary stain is for the endospore stain?
Malachite green.
There it is. Malachite green is our primary stain. Our secondary is that safranin. That's what gave us the red. So the primary stain is, if it's the endospore stain, we're looking for endospores. So we're going to be, our primary stain is looking for the endospores. That was our malachite green. We did use a mordant in the endospore stain. It just wasn't a chemical mordant, the physical one. Does anybody remember how we fixed and the malachite green in? The steam, the heat, yeah, forced it in. I wanted to clarify that because in the endospore stain, it's the steam that forces it in. We had that little paper towel that we sat on top. Remember that? That wasn't forcing this anything. It wasn't doing anything chemically really or physically other than just keeping the moisture on top of our slide. That was the point of it. So FYI, it was the steam that was acting as our mordant there. Our decolorizer was just water when we rinsed it and deionized water. And then our counter stain for the endospore stain was what gave us those red structures. That was safranin. Okay, good job, everybody. So I believe that's going on to a different stain next. Yes, which of the following bacteria would be acid fast positive? All right. One of my other students pointed something out for me, which is going to be the next question on here, which was how to remember the primary stain, which was the F in acid fast. Okay, and we'll come back to it in a moment. But that's your little hint for the next question, okay? If you are acid fast, you have a special molecule in your outside of your cell called mycolic acid, and it is a wax, meaning it's a lipid. So it is hydrophobic, right? Yes, and there's only one genera, which is Mycobacterium. Don't confuse it with anything else. So Mycobacterium species for that one. So there's only 3 genera that you really are going to stand out the most for us in the lab, okay? And that's for now going to be Bacillus and Clostridium make endospores. Mycobacterium have mycolic acid wax and therefore are acid fast. So our next question is, which is relating to our F with the little squiggle under it, might help you remember what our primary stain is for the acid fast procedure. What is the primary stain for the acid fast stain? See if we can recall it. All right, good. I'm glad we kind of picked up on a little bit. F for fuchsia. Fuchsia. In this case, our primary stain is carbonyl fuchsin. Now, if you don't know fuchsia is a color, then maybe that doesn't help you remember. But fuchsia is a color. It's like a pinkish red color. And so here, carbonyl fuchsin is our primary stain. It is lipid soluble. And so it's going to bind with that mycolic acid wax. And so our decolorizer for this one was acid alcohol. It removed any carbonyl fuchsin that didn't stick to the wax. Beautiful. All right, any questions about that? Moving on. Once we get out of the stains, I'll clear the board and we'll remove all this chicken scratch. All right, in which staining procedure did we use steam? You're paying attention. We only used it in one. The endospore stain is correct. In fact, the steam worked as a mordant and helped fix our primary stain, which was malachite green, into place. Next question, which of the following structures allow for bacterial motility? Bacterial motility, allowing our bacteria to move. Now, luckily, your lab midterm will not be select all that apply. Only one will be the right answer. All right, so in this case, this is the flagella. Bacteria do not have cilia, but protozoa can, but not bacteria. Fimbriae are for attachment, and pili are for creating channels between the cells, or we can move DNA between the cells and share our DNA. So we learned about motility, bacterial motility, yes, in unit 1, but also in lab 6 was our motility wet mount. So we won't see a motility wet mount on our lab exam, but we should understand the principles behind it. Remember also about Brownian motion. Brownian motion was the idea that the liquid molecules can move your bacteria around too and makes them look like they're dancing in place, but that's not true motility, only whenever they're propelling themselves. All right, good job. All right, so that's it for our stains. We had the simple stain, which just gave us contrast, the gram stain, which allowed us to tell the difference between our cell walls, the acid fast. Only gram positives can be acid fast. So that's another thing that we will recognize here. When we learned about our Bacillus and Clostridium, also they happen to be gram positive. Mycobacterium are also gram positive. Okay. So if you're gram positive, we might test you for the acid fast. Are you also acid fast if you're gram positive? Only mycobacteriums will be. If you're gram positive, we might do the endospore test. If you're a bacillus or a clostridium, you might be able to make endospores. If you're gram positive, that can make endospores. Staphylococcus aureus, Streptococcus pyogenes, all these other bacteria that are not bacillus, clostridium, or mycobacterium, they're just regular gram positives. Okay. And then there's gram negatives, of course, as well, where they have a thin wall and an outer membrane. The capsule stain, do you have that thick sugary coating on the outside of you that makes it hard for phagocytes to eat you up and also helps with adhesion and building up biofilms? So there's 3 functions of the capsule. I know we had it on our unit 1 test, but be aware that's going to be coming on that midterm, as the lab midterm as well, the functions of the capsule. What else? Capsule. Wet mount, we kind of already talked about motility with that, and the endospore stain, we kind of talked about that. Any questions about any of the stains? All right, and we're going to clear our screen. And we're going to move on to the next part, which I think is the ELISA. Yes. ELISA. The first step of the ELISA is, what does the E stand for? Of course, for the exam, I expect you to know what all the letters stand for. For now, what is E in ELISA? All right, beautiful. I'm glad we've been paying attention. ELISA stands for enzyme-linked immunosorbent assay. And I'm aware that we all know that E is enzyme. So there will be a question on the test where you might have more than one choice that has enzyme in front. So be sure that you know what the LISA is, what that is for, so you can recognize the correct answer for that. Hopefully that one won't trip you up on the test because it's choices. I'm not making you write it out, right? It's just going to be multiple choice. Okay, cool. Remember, there were two different types of immunological tests that you could run. There were direct. I wish this would just click on this. Direct and indirect. That's going to be the next part. So I'm not going to give away too much yet. What is an indirect ELISA looking for? They're both asking questions. The direct is asking us, does the patient have the antigen, AG? And the indirect is asking if the patient has the antibody, which is a way of asking, has the patient ever seen the antigen? So if you guys can remember that a pathogen is the bad guy, right? An antigen is the part of the bad guy that we're anti, okay? So gen is what I'm focusing on there. Antigen or pathogen. Here, antibody, this is a protein made by the body to see the antigen, right? So the antigen is the thing that's foreign. The antibody is the protein that your body makes to see the antigen. All right, so that's how I remember those. B, body, it's made by your body. G, gen, I think it's pathogen. Whatever works for you, but that's how I remember them. Okay, good. You guys are good on that. All right, the principles of how the assay works. It depends on antibody and antigen specificity, right? So that specific interaction between that antibody and that antigen. What are antibodies and antigens? An antigen is what your body could see, your immune system could see as foreign. The antibody is what your body makes, the protein that recognizes that foreign protein, that foreign molecule. Indirect immunoassays, not just ELISA, but any immunological assay. Indirect ones test for the antibody, direct ones test for the antigen. Like I said, if you're testing for the antigen, you're testing for the bad guy directly. You're not asking indirectly, have you ever seen the bad guy? Right, but like literally testing for the bad guy there. So direct is a direct test of a bad guy. How the color change happens, basically it's relying on an antibody, recognizing an antigen. An antibody is linked to an enzyme. If the enzyme cleaves the substrate, we get a color change. The more of whatever you're testing for is there, the darker the color change. So if you're testing for an antibody, the more antibodies, the darker the color change, just like the one that we did in the lab. Rapid testing relies on the same exact principle, but instead of it happening in a well, it's happening on a filter paper. Then as it flows along the filter paper and it comes across a thing, that antibody cleaves the substrate and causes a color change if the thing is there. If not there, it just moseys on its little way and we don't get a color change. So it's seen as a line in a rapid test, but it's the same exact principle. Do we have any questions about ELISA? General concepts here. Okay. Doing good. Check up here, make sure I'm not missing any chats. I am not. All right, on to the DNA stuff. We talked about DNA size, we talked about PCR, gel electrophoresis, and then transformation. So we're going to go over that stuff next. Those all in one lab. All right, PCR, what does it do? All right, so PCR stands for polymerase chain reaction. What type of polymerase? DNA polymerase. All right, so it's going to be copying the DNA or amplifying it. Amplifying just like you would amplify a sound, makes it louder, more of the DNA, amplifying it. Okay, so making copies of it. There's 3 major steps to PCR. The first step is denature, where you're going to use heat to separate the DNA strands. And the second step is the anneal step, and it's probably the most important step. We think of the third step as being the most important, but it's really not. It's this second step. The anneal step, we're going to be slightly cooling, and then the primers will bind. The primers bind because they recognize complementary DNA sequence. Only if the sequence is there will it bind. So this is where that determination will happen. If the DNA sequence is there, the primers bind and recognize it. And we can't have a third step unless the second step happened. So the third step is the elongate. Add nucleotides to the primers and build the other strand, right? So in a sense, we went from double-stranded DNA, and separated the strands. 1, 2, 3, 4, 5. Who cares? 1, 2, 3, 4, 5. Let's do more than that. 6, 7, 8. I need a little more space for the next part. 6, 7, 8. So we separated those strands. That's your first step. That's the denature step. In the second step, the priming step, we're going to prime, which is also known as anneal. We're basically going to take those 2 strands, 1, 2, 3, 4, 5, 6, 7, 8. I should have separated them more. And then 1, 2, 3, 4, 5, 6, 7, 8. It's 1, 2, 3, 4, 5, 6, 7, 8. Get a different color. 1 is going to bind on one side. It matches the A to the T's and the C's to the G's. And we bind our little matching primer. Again, primers are typically about 30 nucleotides. They're not 3, but I'm not going to draw that all out. And then the elongate step, we will match up matching nucleotides, T to A, C to G, and build off of the primer. All right, and then we'll heat it up again and separate those new strands, and then make copies, bind the primers, and then elongate. Separate those new strands, bind the primers, and then elongate. And it's just that that's the whole thing, over and over and over and over again. So that what you end up with, if you had 1 double strand, that became 2 double strands. Each of those became 2. Each of those became 2. And so on and so forth. So in 1 cycle, you went from 1 strand to 2. After 2 cycles, 2 to 4. After 3, 4 to 8. After 4, 8 to 16, and so on and so forth. We good? Okay, so like I said, I'll ask you up to 6 cycles. Okay, all right. Any questions about the DNA or the PCR stuff? Okay. I'm gonna erase this then. I feel like it's pretty straightforward, but also can't go over it enough, just in case. Our next question, which DNA fragment is largest of the list? Remember, look at all of your choices carefully. One of the most missed questions on the lab midterm. Alrighty, you guys did great. It is 35 kilo base pairs because 35 kilobase pairs, oh no, that's not a pen. 35 kilobase pairs, KBP, equals 35,000 base pairs. Good enough? Simple enough? I hope that is easy enough, right? So I'm gonna leave that up for the next question, because the next question kind of relates to that. Which of those fragments would travel the farthest on an agarose gel? Here's our little well that we put our DNA in. DNA has a bunch of sugar phosphates, sugar phosphate on the backbone, each of them attached to some kind of base, nitrogenous base, right? So these P's represent a PO4, and these have negative charges. So negative is going to move to the positive end in gel electrophoresis. DNA is negatively charged, so it's going to move to the positive end, right? As it does that, the longer pieces of DNA will get caught up and the smaller ones will travel fast. We kind of walked through this in the lab, but again, I just want to reiterate that for you. Okay, so the smallest one will travel the farthest. And our smallest one on here happens to be 100 base pairs. So this is where most commonly this question will get asked in this sort of context, like which one will travel the farthest or which one will travel the least far, something like that. So you have to know your sizes of your DNA as well as how it's going to move in the gel. Make sense? Okay, easy enough on that. Right. Next question. We're doing great. Cranking through. What type of cells are capable of taking up exogenous DNA? Okay, so exogenous means that it's DNA that came from outside of the cell. All of these are relative to what we were talking about. No, not all cells ever can take up outside DNA. Only certain types of cells can take up outside DNA, and they are called competent. Can some pathogens take up outside DNA? Yeah, some of them can, but not all of them. And so not all pathogens are competent, and not all competent cells are pathogens either. Some of them are just normal cells. The example that we used in our lab happened to be E. coli, but there are other types of cells that can also do this, not just E. coli. Okay, but yeah, so competent is our bigger term for that one. Okay, before we move on, no, that's fine. We'll go to the next question. It's good. Okay, never mind. We'll go to the end of the DNA stuff. Plasmids and the transformation thing. One other thing that I want to talk about, just a summary of that. That plasmid was P-glow, right? This was a plasmid. It's literally what the P means. It means plasmid. And plasmids are DNA. Literally, it's a big old DNA strand. And we saw the gist of that last time when we did our Cahoots together, right? That it was one big old happy hunky-dory double helix all circled up. And it was, whatever. I'm not going to draw the whole thing. That would take me forever. But you get it. And then on P-glow, we had genes. One of those genes was the arabinose use genes called ARA-C. And that was linked by a operon to GFP gene, green fluorescence protein. And again, these were both controlled by the presence of arabinose, which was a sugar. We had a separate gene on there, which was not controlled by arabinose, which was for ampicillin resistance, right? So this is why if we had just regular bacteria versus bacteria that had a little plasmid in there, these guys that had the plasmid would grow on anything with ampicillin, and these guys on ampicillin wouldn't grow. It wouldn't be able to handle it. So the fact that they grew on ampicillin meant that it had the plasmid. And then if we had arabinose triggered the expression of GFP and Ara-C arabinose use breakdown genes. Remember that one? Okay, so a little bit of a review about that. Yeah. Any questions about the DNA stuff before we move on? Okay. Okay, beautiful. The eukaryotes. You remember this little thing where I showed you our cheat sheet with our stains? I want to show you something else on it. All those pictures that we took. This was here the whole time. But hey, okay, there's a difference though. I know, but there's a difference, okay? Dipylidium. Our dipylidium, ours were pink. Ours were bright pink. I don't know why, whatever, bright pink. We'll lean into it. It's cool. Ours were bright pink. So I took these pictures, literally copy and pasted them from the internet. I haven't updated these in years. So I have new pictures, but ours were pink. Our dipylidium were. So that's a big difference. They look like big clusters of pink grapes, those ones did. And our Clonorchus was way more zoomed in than this one too. But so anyways, but other than that, our Aspergillus was more of a teal green color. And that's actually how I remember the difference between Aspergillus and penicillium. We only had to look at 2 fungi. Aspergillus and penicillium. P for penicillium is pink to me, or purple. If that's purple to you, don't let me stop you, but I think it's pink. P for pink. Aspergillus, G for gillus, is green. To me, it was a teal green. It wasn't the same color as this. Ours was greener. Okay. You notice here on Giardia and Entamoeba histolytica, I put who cares, not you. Because we're not going to have those ones be the answer on the test. They might be a choice, but they won't be the answer. Because they're hard for me to find. They were hard for you guys to see in the microscope. So I tried to pick ones that were easier to see. Okay. So they will not be the answer. It might be a choice. Just FYI. Your answer for the protozoa. So we'll have a fungi question. And I'm going to tell you that they're fungi. The protozoa one, you're going to need to know it's protozoa. I feel like the eggs looked like the eggs and the protozoa looked like the protozoa. They didn't look the same. And you cannot tell me that Trichomonas and Trypanosoma looked the same. They look wildly different than one another. So you got the weird purple snakes. They're purple snake looking guys on ours. Ours was tinier, but that's zoomed in more. Versus these guys, which if I zoomed in on this, which I could download it and zoom in, but you would see that our Trichomonas guys, they look like this. They're like kind of a light color fill in. They've got like a big old nucleus and they've got a little tail coming off of them. Completely different than the snake shaped guys on the other image. And ours were purple. It was purple all over the place. Like it was very clearly like all over the place. Versus the snake guys on the Trypanosoma. What are those little blobs around the snake guys? Do you guys remember? Red blood cells. So knowing that also put that into context. They look drastically different than one another. So that should be helpful. For the eggs, focus on the major differences, okay? And you will be fine, I promise. Okay? Okay. So now that we have gone over this is there. For the eggs, some of them were bigger than others. Some of them they have different shapes. So to me, when I look at this, look at Taenia. It's more round with round inside versus Ascaris. It's like round with little guys inside, like more bubbly inside. And Pterobius, oval. Clonorchus, ours was way bigger. It was big and it looked like this. It had a little like gutty works inside. Yeah, it had little, I kind of say gutty works, but yeah. Yeah, it looked like, yes, it did. It looked like papaya. And that was where the eggs were inside of it. That was like their egg pouch or whatever. And then our schistosomes, they had these little spikes coming off of them. Our dipylidium were bright pink, like grapes almost, okay? And so we can already see that they're all different Your hardest difference is going to be between Ascaris and Tinea. That's your hardest one to know the difference between, okay? And I will make sure that whatever one you're looking at, that will be clear what you're looking at. Okay? You will have one, not all six, one of them. You'll have one protozoa. You've only got two you really need to know. Okay? One fungi, you've only got two. And then the eggs, one of the eggs. There's six, but they are different. So focus on those differences, okay? One thing I want to point out with the fungi, a different color. Didn't mean to click that. What are those structures called? They look like penicillium. It looked like little paintbrushes. And then the aspergillus looked like dandelions. Again, if I zoomed in, you'd be able to see this. These are called conidiophores, not sporangiophores. So conidiophores or conidiospores, I would accept either as conidio. I won't put conidio anything else. Conidio, okay? Why are they called conidio? Because they're the open ones. The sporangio ones had a big covering around them and they would burst open. Remember that from unit 1? Okay, so that's come back and I will ask you about it on the test. What are these structures called? Okay, conidio. All right, are we ready? Okay, so we're gonna go back to the questions. Yeah, so we can see here, this would be a drawing of penicillium conidiophores. Right, which organism is this one? And it is the same color as yours would be on the test. Excellent, that's penicillium, pink for penicillium. That was the fungi, that was the conidiophore. Beautiful. Next one. What is the name of this organism? On the test, I'm more likely to put other protozoa names next to it. FYI. But for reference, this was Trypanosoma, our purple snakes. Our other protozoa that we're going to have to know will be Trichomonas. Okay, so that in consideration. Enterobius was one of our worms. We need to know the eggs for those ones. Rhizopus was a fungi. We didn't need to know what it looked like. Okay, syphilis is a bacteria. Just so we know, those would also be relative to those organisms and species that are. Okay. You guys are doing great. Recognize your organisms that it says that you had to draw. We took pictures of them. So recognize those. Important structural and reproductive differences, as well as just visible differences. Focus on the differences there. The concept of a trophozoite versus a cyst. With our bacteria, again, I know I'm talking about bacteria, but hold on. And this is the eukaryotes, but hold on for me for a second. With bacteria, the dormant guy, the dormant and inert resistant guy versus the active guy. In bacteria, if you made the dormant version, they were called endospores, right? The active ones were called vegetative. In protozoa, again, this applies to protozoa, so you might want to know that it's protozoa that do this. But the protozoa, the resistant ones are called cysts. The active forms are called trophozoites. And I literally remember this because troph. Now, chapter 9 goes through a lot of different kinds of trophs, but they all have to do with like eating and nutrients. You only need food if you're active. And that's how I remember it. I don't know if that helps you, but it helps me. So anyways, that's our comparison there. All right, so I might ask you about that. Focus on your differences between the images. Oh yes, and the transmission. So I did again, review the transmission stuff with you. Complete the following table: transmission routes and definitive hosts. I will not ask about definitive hosts, but I will ask about a transmission route. Okay, so that was on that worksheet for that. We okay? So I've narrowed it down a little bit. Okay. And yeah, I know it was there the whole time. Don't cry about it too much, okay? Okay. Any questions about the eukaryotes? I feel like it's pretty straightforward. Next, we'll talk about bacteria stuff, general bacteria stuff. So this gets into cell shape, cell arrangement, and colony morphology and the different types of media, as well as isolation. We're almost there. Which of the following is not a term used to describe colony morphology? Okay, so all of these terms, convex, irregular, punctiform, rhizoid, plateau, those guys have to do with colony morphology, what you see with the naked eye on the plate. There are some crazy terms that we use for colony morphology, like punctiform, which just means the tiny little dots are the shape of your colony, the tiny, tiny, tiny little dots. Punctiform, umbonate, pulvinate. These are all terms that relate to colony morphology. Be aware that they are colony morphology terms. I will not ask you to define them. You're welcome. But know that they are colony morphology terms. Some cell shape and arrangement. If we're talking about cell shape, we'll be talking about the bacilli. Bacillus is single. Coccus is singular term, right? Versus arrangement. This would be Staphylo or Strepto, or no arrangement. I know that there's more than this that exist in the world, but this is all I have in the lab of the organisms that I have. So we'll break it down even further. Strepto is the chains, and Staphylo are the irregular clusters, or bunches, however you want to look at it. Bacillus versus coccus. Coccus is round or sphere or circle, whatever you want. C-O-C-C. Those are all really round letters, and that's how I remember that. Whereas bacillus, the L's look like the shape of the organisms, rod shaped. It helps with memory, okay? Any questions about the general idea of this stuff before this? Umbonate. Sorry, my handwriting is terrible. Umbonate. It is like fried egg shape. Yes. I'll rewrite it because it was bad. You're not wrong.
So is my eyesight.
Okay. Okay. So now we're going to go into some questions. This isn't going to get everything, but it's a good starting point for our review. Okay. What is the name of this type of culture? Okay, good enough. So I reviewed stuff that didn't even come up first. That's fine. Talk about this stuff too. All right, there are 3 major types of solid media that we need to know and 1 type of liquid. Here, if we looked at the image that we were shown, this is assuming that these are standing upright. They're not tipped on their side or anything like that. They're standing upright. They are solid and look like that. So that's the slant. It is that easy. It is meant to be. Okay, those are easy. It should be easy. Don't overthink it. Okay. still say that. Where is my, oh, it went up here for some reason. Whatever. So our different types of solid media, we have the plate, we have the slant that we just saw here, and we have the butt. We haven't worked with a butt yet, but we will in the second 8 weeks of the course, okay? They're basically like, you know how your tubes look when they're liquid, but imagine it was solid, like a flat in, like it's like in a tube, like a liquid, it's flat on top, it's not at a slant, okay? Liquid is broth, easy enough, okay? If you were made solid, we added something to it to solidify it, and that is agar, which is from algae. It's a solidifying agent. Solidifying agent from algae. I just wrote it weird, but yeah, makes sense? Okay. Okay, good. I think we're ready. Let's go on. How was the organism in the image isolated? By what method? Okay. I'm going to go through all of these, but the one that you really need to know is the streak plate, which is one we did in class, which is why everybody recognized it. The streak plate, also known as the quad streak plate, we would take it out on a plate and we put the first quadrant, the second quadrant, that's a 2. The third quadrant, go into it and zigzag out, and then the fourth quadrant, zigzag out. Remember this? Okay, so the whole idea is that in each one, we're going to flame our loop in between each one to help spread out the cells. It's a physical dilution. But when we talk about actual dilution, like in serial dilution or loop dilution, we're doing this in a liquid. Well, in the case of serial dilution, we're taking a certain volume and adding it to another larger volume. In the case of loop dilution, we are taking a sample on the loop and then mixing it into a large liquid volume. But they're both involving liquid. To dilute it out with liquid, like we typically think of diluting being with liquid. That's how we'll use that term. So spreading out the cells on the streak plate. I am not going to test you on anything except the streak plate. That's what we did, it's what we're familiar with, but FYI, Unit 1 did talk about these other methods, just so we're clear, okay? Now, this is not your Unit 1 test, this is your lab midterm, but that doesn't mean it can't come back on the final. So that's why I didn't want to leave it alone, okay? Okay, any questions about the streak plate? Which quadrant will have the most isolated colonies? 4, excellent. Okay, I'm just gonna erase this stuff. I don't want to get rid of all the good stuff. All that fancy, colorful stuff above it. Bear with me. Okay, I'll erase this when I get to the next question. Next question, which arrangements of bacteria do you observe in the image? One thing I want to be very clear about for the lab exam is if I'm asking for the cell shape, I'm asking for the cell shape, not the arrangement. I'm asking for the arrangement, I'm asking for the arrangement, not the shape. So don't get them confused. Don't just try to regurgitate everything you see on there either. Okay, so the overall arrangement of this guy would be strepto. And I know we are tempted to look at this clustering right here and be like, that looks staphylo to me. Those are quite clearly chains that have bound up back onto each other. And when I talk about arrangements for the lab exam, I'm asking about the overall arrangement. Might you see 2 stuck together and want to see, oh, that's diplo. Is the overall arrangement diplo, or does it just happen to be 2 stuck together on the side over here? So the overall arrangement of what you see in the slide. Now you notice up here on you guys up here, but you notice how I have staphylo, strepto, or no arrangement. I do not have any samples that aren't one of those. So you won't see any like sarcina or anything like that. It will be staphylo or strepto or no arrangement. And I will make sure that I pick one that is obvious. Okay. Same thing with the cell shape. If you sat at table 3 in the lab, you might remember that your gram-positive is a coccobacillus. Well, on the exam, I'm not going to put a coccobacillus. I'm going to put either a bacillus or a coccus because I don't want you guys getting confused about it. Okay. So focus on those there. I don't have any other shapes. I don't have some of the other ones that like we talk about in unit 1. Okay, we're going to move on. Next one's going to ask about shape. What is the name of the shape of the bacteria pictured? And for you guys on the exam, it'll be like, what is the name of the shape of these bacteria? And it'll be a slide that you'll look in the microscope to see. There's that word, Brianna, the umbonate. That's the word. Okay, so umbonate means fried egg shape, and it is a colony morphology term. So it's not a cell shape or arrangement term. So it doesn't even belong here. One of these things is not like the other, and it's umbonate. For what we saw in that image, these are quite clearly rod-shaped cells. These are bacilli. Cocci would be round, and spirillum would be spiral-shaped. Again, we won't see them on the test, but that doesn't mean it won't be a choice. Okay. Good. All right, so be aware of your colony morphology terms. Again, be able to recognize that they are colony morphology terms or cell shape terms or cell arrangement terms. You'll have to identify cell shapes and cell arrangements, okay? Isolation techniques. Be aware of your quad streak method and know that your isolation will happen in quadrant 4. Yeah, we covered everything here. Okay. Last part. Microscopes and knowing how to use them properly. There's no part on the test where I'm going to make you set up a microscope and find the thing or anything like that. But you will have to know that it should be clear when you look into it, and you'll have to know enough to ask me to focus it or focus it yourself. You are welcome to use the fine adjustment knobs if it is a little blurry, or if you want to try to see if you can adjust the focus for your preference. Fine adjustment is fine. But if you get to a microscope and it is blurry and you aren't comfortable messing with it, then just call me over and I will adjust it for you at no cost to you. Okay, so if there's ever a case like that, but you should be able to see whatever the image is. Okay. So know your parts and know how they work, because that will be a question on the test also. For magnification, if you observe a specimen with a 40x and you have a 10x ocular lens, what's the total magnification? Okay, 400x, we're going to multiply them together. So if you're multiplying by 10, you just add a 0 on, for example. Okay. So that's the idea. So if I set up a microscope, no. And here it is. Microscope. Why did I have to start drawing it like this? Lens. Here's your lens. Here's your stage with your sample on it. Here's your light source coming up out of the light source. Here's your base. Here's your nose piece. This is not bad, actually, now that I'm... Here's your adjustment knobs. And then here's your eyepiece coming out here. And then here's your little eyeball looking at it. Crazy, I know. Okay. So what you see in here, this is always 10x. And then your lens, whatever lens you're on, will have a value on it. Let's say that this one was, in this case, for the example, 40x. If that 40x is what is on your slide, then that is what you will use for your multiplying. So on the exam, I will have a microscope set up. It might be even one that has other questions for it. And the question might ask, what is the overall magnification for this microscope, how it's set up and how you're looking at it? Like the image that you're seeing, how many times is it magnified? The idea here is that you would look and say, oh, she has it on the 40x. The eyepiece is always 10x, and it says it on there too, in case you forget. So you just multiply them and you get 400. Make sense? If it's on the 100, then it would be 1,000. We good? Okay. That's the idea of how that would work. I'm gonna leave this up for now. Next question. Which objective lens do we use for immersion oil? Oil immersion. Wonderful. Okay, we all know it's the 100x, at least we're on that together. And what is the purpose of oil immersion? It reduces light refraction. Light scatter, also known as refraction. This is because the oil is more similar in density to the glass than the glass is to the air. So if we go from light to glass to air to lens, we lose a lot of light scatter and therefore a lot of detail in our specimen. In fact, we actually may have seen that. If you guys ever switched to your 100 without the oil, it was really hard to see the organism. I think that oil really made that a lot easier to see that because we're retaining those lenses, okay, or those light rays. Know the parts of your microscope as well. Here's some examples, or let me wait till we get to this. Okay, there's not examples. Let me come back. All right, this part, a different color here. This part here, this part versus this part, or this part, or this part, or this part. Each of these that I made look like udders hanging down. Each of these are objective lenses. So in this case here, that is our objective lens. But this part that the objective lenses are attached to, ours is black. It's a black rubber thing, and it's what we use to rotate between the lenses. You guys know what that is called? It's called a nose piece. Maybe you know that. Now, I'm not saying for this test necessarily. We got 2 lab tests, a midterm and a final. One of them will definitely ask about the nose piece, and for some reason, students like to argue with me about it. I'll put a piece of tape on that black rubber piece, and that'll be the question, what is this part? And then what does it do? 2 questions associated with it. And I had multiple students argue with me, and I have a tape on that black piece, and I'll be like, that is the objective lens, and you can't be wrong. If you don't know the difference between the objective lens and that black piece, I don't know how to help you. And if you didn't know that the black piece had a name, that's not my fault because it is covered in the lab manual. And I have talked about it multiple times in the lab. So I just want to be sure we're covered on that. So that if you see it again, it might not be on this test. It might be on the lab final, but it will be on one of them. FYI, it's a nose piece. Okay. And those things that hang down are called objective lenses. Okay. When we're creating the image, the light travels like this through the microscope. Starts here, it comes up through the specimen and hits your objective lens first. This is which image? Do you guys remember? The real image. Then it travels up. Actually, let me change the color of it because I think that'll be easier to see it. Let's do green. Let's do this lime green. Then it travels up through some mirrors and stuff like that. There's mirrors and stuff in here. And it comes through this other lens here to create the second image, which is called the virtual. Virtual image is made at the eyepiece, also known as the ocular lens. The eyepiece is what the physical thing you look through. The lens itself is the ocular lens, right? The real image is made at the objective lens. Based on the light travel. The light travel hit that objective lens first, and then we changed it a second time and made the virtual image. Okay, that's why that is the way that it is. I want to see if there's anything else I'm missing. Okay, seems good. Any questions about microscopes, their parts, or how they work? So I think we're at our last question. The Kirby-Bauer assay. So looking at this image, just before we even get into questions, this image is not the one we're going to use for the questions. I would say for this, I don't like that. Hold on, darker color. Pink is probably better here. Yeah, you can see it better. For this one, I would say resistant. I would even say for this one, resistant, right? Look at all that growth in there. Quite obviously not. It's inhibiting it some, but it's still growing some. So I wouldn't use it. I'd probably even say that this is resistant and this is resistant. All of these other guys are susceptible. Why? Because they have clearings around them. I mean, this one's a little bit questionable. I'm not going to ask it on that one on the test though, right? This one's either a little bit susceptible. Not my top pick though. All right, so this is the Kirby-Bauer assay. Now imagine I take a little sample in this clear area and move it onto a new plate. If it grows, so if it grows versus it didn't grow. If it grows, then this antibiotic, whatever it is, if it grows, then that antibiotic was bacteriostatic. If it didn't grow after we sample that clear area, then that is bactericidal. We're tracking because we know in this area there's no growth. We know that, but we'd have to culture in the clear area again to determine if it's bacteriostatic or bactericidal. So this is where we got into that review from last time for the Kirby-Bauer, that if you are doing Kirby-Bauer and you have an antibiotic, you are either resistant or you are susceptible. If you are susceptible and you had a zone of inhibition, if you had a zone of inhibition, then if we check in the zone of inhibition, if you grew, then you are static. It's fine. Or cidal if you didn't grow. So in the case of static, we just had no growth. In the case of cidal, we actually killed the bacteria. Are we tracking that okay? Okay. What is the name of the media that we used in the Kirby-Bauer assay? It's not called Kirby-Bauer media. Mueller-Henson. Mueller-Henson agar has a set controlled pH, it's pH controlled, and it has a set controlled depth. Both of these work together to facilitate antibiotic diffusion and stability. Do you guys also remember about how we can measure that zone of inhibition? So we know that you measure that zone of inhibition completely all the way across the diameter. That's the clearing area. So you can measure that zone of inhibition there, but you can use that to calculate the minimum amount, the minimum concentration needed to inhibit growth. We call it the minimum inhibitory concentration, the MIC. All right, just be aware of the concept of it. All right, that's it for the concepts for Kirby-Bauer. This is wild. I just drew a whole ton of R's and S's on here, so I'm going to erase some of these. But we're going to go on to the question regarding this stuff next. I'm going to leave some of these ideas up so you have some reference. Wish we could just like erase part of this. Okay, so we're going to go on to the next question. This is the last question. Okay. Which antibiotic in the picture here is this bacteria least susceptible to? Least susceptible. I'm glad we were mostly in agreement. Remember, susceptible just means sensitive. Least susceptible means most resistant. Most susceptible means least resistant. So in our image on that Kirby-Bauer assay, which one were we the most resistant to? E, also known as least susceptible. Make sense? Okay. Any questions about this? Okay. Talk about ZOI as the zone of inhibition. All we know on that Kirby-Bauer plate is that it didn't grow there. Until we do the second test, we don't know if it killed it or if it's static, right? Yes, since we talked about Mueller-Henson and minimum inhibitory concentration. I'm not going to make you calculate anything with that, right? Just be aware of what it is. All right, that is it. Okay, so if you have any questions, just email me with any of your questions anytime before the exam. The exam is on Wednesday. Be prepared with your lab coat and your closed-toed shoes. Otherwise, I'll go over all of the other lab rules and walk you through the test and everything whenever we get into the exam on Wednesday. We've walked through it once. We have an idea of what to expect. It's all multiple choice. It's on your laptops. Be sure your laptops are charged. For some reason you have an issue with charging your laptop, just let me know and we'll give you a paper test. Okay? Any questions? All right.
So the test, we bring our laptop.
Yes. Mm-hmm. It'll be given the exact same way as the lecture exams were. So on the Canvas lockdown browser, and we'll walk around with our laptops. Yes.
Oh,
Yeah. Yes. Yes. So we have on announcements. This is my view. So you guys don't have the same view as me. Let me see student view. I'm looking at this. I'm like, oh, these are all future announcements. Sorry. Okay, so on your announcements, if you have lab midterm review Kahoot, it'll have this Kahoot, which is at the top, as well as all of the lab Kahoots for the lab. Yes.
I was wondering, how can you redo the one on the bottom?
Use a different username for it.
Access for your
Email.
and it won't let you do it.
Okay, use a different email. If you don't have a different email, then I don't, yeah, exhaust your emails. Yes, so that's the way that it's set up, like the way they're set. Some of them on the online ones, these ones, I mean, if they ask you for the emails, I used to have them set up where they didn't. So that was, I don't know why I had to ask for it now, but
I didn't know.
yeah, no, it doesn't have to be any email. And just make up an email, honestly. Use your husband's email, use your boyfriend's email. Yeah, do this, yeah. Use a different email. Yeah. Make up an email, get a new Gmail address just for this. I don't know. The more you want to play it, the more emails addresses you're going to need. But yes. So if you get a different email address, like your home email address or something, I'm not going to double check it or anything. I'm not like picking off of you guys' emails or something. Yeah. So just do that. And then you guys can like walk through and just make sure that you are, copying what you need out of them, make your own Quizlets or something out of them. That'd be fine too. So yes. Yeah, I didn't realize I needed you guys emails on them. I didn't mean for it to. But yeah, so yeah, you have that available as a resource. Remember, you have those study sheets that I showed you where they were, and you have study guides to help you work through like the concepts. And there's plenty of resources on there for that. Yes. Yeah. Have you been making yours?
I did too.
Okay. Yeah. So if you're somebody like, like I said, if you're just wanting to knock out your lab concept maps and get points for them, don't let me stop you. But if you're sitting down and actually walking through the lab concept maps and making them consciously, which most of my students do, I'll be honest, most of them do. But if you're making the lab concept maps consciously and writing it out from your understanding, then I feel like that helps a lot, just like organizing the thoughts with it. So if you haven't done that, then I recommend it. And you have until basically the lab exam to knock them out. So anyways, yes, do you have a question, Alvin?
How can I get started with this?
To this, it should be in your announcements on Canvas. So it'll be called Lab Midterm Review Kahoot. All right, any other questions? Unit 2 is the following Monday. Yes. For most of you guys, yeah.
If I had an issue with the babysitting, like I told you,
Mhm.
we can figure out.
Just let me know. Just email me. Yes, yes, absolutely. There shouldn't be a problem. As long as you come to one of them in person, it'll be okay. Yeah. All right, any other questions? Okay.
Professor,
Yes.
I have a question. Did you say that we can turn in our lab concept maps for extra credit and up until the exam date, or were those due on Sunday?
What did it say on the thing? I don't even remember. Was it Sunday?
It was due as of the 7th, so not yet, tomorrow.
Okay, tomorrow at midnight then, I think, right? I think that's what it is.
Okay.
I see. Yeah, I'm like, now you're asking me and the questions that I have to look this up. I'll double check it for you right now. Pre-lab, in-lab, post-lab. Connect, extra. Okay, here it is. October 7th by midnight. So tomorrow by midnight for all of those. Up to lab
Okay.
11 or 11, as many as you want, as many as you get done. It's, you know, one is better than none. So yeah. Yep. Any
Okay,
other questions? Yeah, absolutely. All right. Thanks, guys. I will see you on Wednesday. Good luck. Happy studying. And I will post this recording online. FYI.
Regular lecture concept maps, for the labs kind of follow the objectives as well.
Yes,
Okay.
yes. There's learning objectives. I'm not as picky about
Okay.
if you're covering the general concepts, then the fact that you're doing them, I generally give credit for it.
Oh,
Yeah.
okay.
Yes.
Like they help me out studying when I did those